So good morning everyone. In this video I am going to discuss about most commonly used steps in the docking studies using any kind of software, whether you are going using commercial softwares like Mesh Pro, Biological So, Discovery Studio, or Autodoc, which is freely available. The steps to perform a docking study is somewhat similar. As I told in earlier lecture also, to perform a docking studies, you should have two things. One is you should have the structure of the molecules, and another things you should have the structure of the target. Target can be anything, so your enzymes, your DNA, RNA, or proteins, depending upon your requirement. But to perform a docking study, you should have these two things. So first thing is, if you know you have to draw a structure of the molecules, and most commonly used tool is ChemDraw, by which you can draw the structures of the molecules. There are also option in the ChemDraw where you can put the molecule name. If it's already available, automatically the structure will reflect. Next step is you have to prepare the structures of the molecule which you have drawn in the ChemDraw. And preparations of structure means you have to do the energy minimizations, and energy minimization means you are converting that molecules into a most stable configurations. So that you have to do energy minimizations of these molecules, or you can generate a conformers depending upon the softwares. Next is target, from where you will get the structure of the target. You can get the structure of the target from the PDB, that is a protein data bank. Just do the Google's PDB, type the PDB, you will get the websites of protein data bank. In that one, in the search items, you can put the desired target, and then you can get a structures of these targets. So there are steps by which you have to do the selection of target depending upon experimental condition, depending upon your R square values. So you have to be select the most appropriate target for to perform a docking studies. The next step is, after that you have to import the structure of that target in your software, whatever the software you are using. In many software there is a direct options. If you have internet connection directly in the software, you can put the PDB ID and target will be available in the workspace. So in after selection of target also, you have to be do the preparations of target. Preparations of target means you have to be add the hydrogen ions. Hydrogen ions we have to be add to perform any docking protocols. The reason is because hydrogen ions are missed in the NMR or in X-ray crystallographies. So you have to be added before doing a docking studies. In most of the protocols, we are removing the water molecules. What is, it depends upon the nature of target. If your target is of GIT track or something where the actual environment is water, so you have to there is no need to remove the water molecule. Otherwise you have to remove these water molecules, and then you have to do the energy minimization as you have done for the preparations of the structure of the molecule. So you have to do the energy minimization. Again, this is the most stable configurations of that proteins. And then now you have prepared the structures and also prepared the target. So next step is you have to be generate a grid, that will be called grid generation. Grid generation means you have to be generate a active site of that particular target. See, the molecule is not going to bind with the whole targets, it's going to bind with the active sites of that particular target. So the in computers you have to be tell the computers where your active site is. So there are the different methods by which we can generate the active sites. There are some proteins molecule have internal ligands, so by using computers you can tell the internal, where the internal ligand is present, generate the active site there. If it's not available, internal ligand is not available in the protein, you can predict the active site amino acids. So you can specify the residue in the software. These are the residues whose makes the active site or generate active site in these residues. Another thing is, there are in many software there are a tool of site map analysis where you can predict the sites in the target, right? What step is, you should generate the active site, that we call grid generations, before performing the docking protocols. If you are not predicting the active site, then you can't perform a docking studies. So docking studies is rigid or flexible. Most commonly used is a flexible. Flexible means your molecules and protein is flexible and it's somewhat close to your real environment. So flexible is most preferred, but yes, it will take much more time as compared to rigid. There are a different when you perform a protocol, it will ask whether you are going to perform a high throughput screening or standard precision or extra precision. High throughput screening you have to use if you have a large number of compounds for the screening purpose. So we have to use high throughput screening protocol. If you have a 100 molecules or 200 molecules, you can perform a standard precision. And if you have a less number of molecules, then you should perform the extra precision. So these are the methods by which you can perform the docking studies. And last is after performing the docking study, you get a results, and you have to be select best docking scoring function. So we assume that the more the negative, the more the stable configurations of that particular compound in the active site. So we are assuming that compound is more have more stable configurations in that active sites. In some cases, we are comparing the results of our docking scoring functions with the standard molecules that we call internal ligand. Internal ligand is a molecule which is already available in the protein in the active site, so you can predict yes, your screened molecule is how much close to internal molecules. So these are the common steps which we have to be performed before means to do any kind of doctoring studies or to use any kind of softwares. So I hope you get an idea. Thank you.